Hello, my name is Timothy Francis. I'm the Senior Technical Specialist for Fujifilm Wako Chemicals USA Corporation, and I'm excited to be providing this portion of this presentation on adapting your laboratory microplate reader for bacterial endotoxin testing with molecular devices. I'm going to be presenting to you an overview of the bacterial endotoxin test, as well as some of the studies we've done with the reader in the performance with our reagent. First, I'd like to talk about what the bacterial endotoxin test is. Here we see the USP 85, the bacterial endotoxin test, is the title of this slide. USP chapter 85 outlines for us the bacterial endotoxin test with what is called the Limulus immunocyte lysate reagent, which is what our company manufactures. There are three techniques listed in USP 85 for the bacterial endotoxin test. Now this chapter is also harmonized with the European pharmacopoeia and Japanese pharmacopoeias as well. The first methodology is called the gel clot technique. This measures endotoxin by the presence of gel formation after cleavage of peptide bonds within coagulogen to yield coagulin. You can see the LAL cascade that I've illustrated here, showing the reaction that takes place with these protein factors when endotoxin is introduced. So the gel clot technique is literally just reacting LAL reagent with a sample in a test tube after incubation of 1 hour. At that point, the test tube is inverted and a positive test is recorded if that solution is clotted and remains at the top of the test tube. At that point, we would say that that sample has endotoxin, at least at the labeled sensitivity of the reagent. You can see this is a qualitative method. It only gives a positive or negative result at a specific sensitivity. The turbidimetric and chromogenic techniques, however, are quantitative methods in that they will allow a standard curve to produce definite endotoxin results. Now, the turbidimetric method has a measurement principle very similar to the gel clot method. In fact, our reagent is licensed to perform either method. But the turbidimetric method involves an absorbance reader measuring the onset of turbidity of that reagent. In contrast, we also manufacture chromogenic reagent in which a chromogenic substrate has been added to the reagent to measure the onset of this clotting enzyme more effectively using an absorbance reader. So these are the three general methods that the USP 85 outlines in the bacterial endotoxin test, and we're going to be looking specifically at turbidimetric and chromogenic methods. Now, we're going to look at what the USP 85 requirements of a quantitative method are, specifically of the reader itself. The reader needs to be a photometric incubating reader. It needs to be capable of measuring a negative control without activation. This may be stating the obvious, but of course a negative control is supposed to be there to ensure that the environment is free of contamination. So only activation of the negative control should come about by the sample that we are attempting to measure, in which case would be endotoxin. And then whatever regression is used for the standard curve regression, the correlation coefficient must be greater than or equal to 0.980. Or of course, if it's negative, it should be less than that. Basically, the absolute value of that correlation coefficient should be greater than 0.980. Samples must be tested with a positive product control spiked at or near the midpoint of the standard curve, and then measured positive product control results must lie within 50 to 200% of a true value. Now, many errors, whether that's technical, user, environmental, as well as product interference itself could all come into the variability of the assay. So this 50 to 200% range you can think of as the resolution of the test, but in a sense it's kind of that catch-all variability we allow for this biological assay. Now all of our reagents are kinetic reagents. We measure them using kinetic methods. There are endpoint endotoxin reagents out there in which incubation takes place over a set amount of time and then one measurement is taken. However, we use kinetic measurements for both our chromogenic and our turbidimetric techniques. Some of the advantages of this are there's no need to take the time yourself, at which point a plate would need a stop reagent added and then a measurement taken place, as well as a kinetic method allows for a wide standard curve range. I visualized that here for you. First, you'll notice the negative control remains unreactive above our threshold OD optical density. That's set at 0.015 for our reagent. Basically, that's the threshold at which an activation time is recorded. You can see I have 4 concentrations ranging from 5 to 0.05 EU per ml. That's just endotoxin units per milliliter that have an activation time. So we have a measure of concentration that gives us a measure of activation time, and from those values we can create a standard curve. Now, any regression is allowed by the pharmacopoeial standards, but our reagent specifically, we use a log-log linear reduction for the regression of that standard curve. So you can see that here. Basically, on a logarithmic graph, you see the CSE concentration on the x-axis, the onset time on the y-axis, and you can see the absolute value of linear regression that we received from that. So that's an overview of the kinetic measurement, and then based off of unknown samples activation times, we're able to plug those values into the standard curve to get measures of the endotoxin concentration. I now want to give you an overview of our kinetic endotoxin testing reagents that we provide. First of all, the Pyrostar ESF plate reagent. This is a kinetic turbidimetric reagent, and we validate it to produce for you a standard curve from 10 to 0.01 endotoxin units per milliliter. We also manufacture the Limulus Color KY reagent, which is our kinetic chromogenic reagent. You can see one of the advantages here is that the quantitative range is from 5 all the way down to 0.0005 endotoxin units per milliliter. Now both of these are Limulus amoebocyte lysate reagent. Both of these fit the definition of an endotoxin testing reagent according to USP 85. However, the United States Pharmacopoeia has just voted to include a new chapter on bacterial endotoxin testing. That's chapter 86. And this chapter allows for synthetic reagents to be used. So I've also included on here our Pyrostar Neo reagent, which is a completely synthetic reagent, but it's measured by the kinetic chromogenic technique. And in fact, all of the settings are applicable among those two reagents. It has a quantitative range from 50 to 0.001 endotoxin units per milliliter. Now the USP, as we mentioned, has some requirements for quantitative methods. However, we as well as the reagent manufacturer have set additional requirements in our package inserts for accurate measurement with our reagent. So we validate that our reagent will provide for you results that are within specification if you meet the testing parameters that we require for valid plate reader measurements. So you can see those listed here for our kinetic chromogenic and kinetic turbidimetric reagents. nearly identical requirements for each. Incubation temperature of 37 degrees Celsius throughout the read. Of course, assay mode of kinetic. I mentioned that threshold optical density of 0.015. We ask that you perform a shake at the beginning of the protocol before the reads start. The shake often we recommend around 10 seconds on a medium constant speed. Measurement wavelength is where these two methods do diverge from each other. Both require a measurement at 405 nanometers. However, the chromogenic reagent also takes measurement at 650 nanometers, and this signal is subtracted from the signal taken at the same time from 405 nanometers. This is to ensure that all of the noise from the general turbidity is subtracted and only the chromogenic portion of that signal is measured at that wavelength. We also recommend, if your reader has a carriage speed setting, that be set to slow. The measurement interval is 40 seconds for each of the reads, and then for the standard curve measurement criteria, that log-log linear reduction that I mentioned. So our package inserts outline these points. If you follow these points with a valid microplate reader, then we guarantee the results of the reagent. Now, what we have done is we've partnered with Molecular Devices to create a protocol for you that fits these criteria, so no need to set that up. It's automatically set up for you in the protocols. So what I want to do is look at some example data of a comparison of an assay run on two different readers, the molecular device reader and a reader that we would use in-house that we currently use for the validation of the reagent, to see the differences in the data, in the similarities, and also the validity of the report as it is generated. So first, I have data using our Pyrostar ESF reagent, which is that kinetic turbidimetric reagent. We ran the standard curve along the full range that we have validated the reagent at, from 10 all the way down to 0.01 endotoxin units per milliliter. We've set up the reagent with the parameters that we would have specified in the protocols. And you can see the activation times and the linear regression equation that both readers would have produced. Now, we'll get to the measure of validity in a moment in the specifications that USP 85 requires of a valid standard curve. But I do want you to notice at the offset here, we are getting slightly different values for activation time. Now, you'll notice I haven't mentioned any specific requirement for activation time, and I'll explain the reason that's the case is because there are so many variables that can affect activation time, but are accounted for by the production of a standard curve. But we'll take note and I'll provide an explanation of why these differences are reflected, and you can see that slightly in the standard curve. So they're very similar values, but there is a slight lengthening of the numbers for the SpectraMax ABS plus microplate reader than this reader that I used, that we use in-house for the validation of the reagent. When we look at the data that was produced, we'll start to understand the slight difference in activation times. First of all, I want to mention the fact that the linear regressions for the two are very, very good, nearly identical. So we have nearly identical correlation, and of course, over time, the averages, we'd see no difference between these. But one thing I do notice, and it may be hard to see on the screen, is that the the SpectraMax ABS Plus reader is showing a different value for the optical density of the final reaction that takes place. That's perfectly fine. Once again, we have no specified values for that. But what that does mean is that that measure of optical density is relative. It is unique to each of the readers. And so that reason right there with that threshold value of 0.015 is the reason we got slightly different activation times. However, we can, in a sense, cross-reference calculated values into the standard curve to eliminate some of these variables that would have come about by the optical density. And I show that right here. So basically what I have in the first graph is a measure of the true values in the terms of the control standard endotoxin concentrations that were used, and the values that were produced by the in-house reader and the SpectraMax ABS Plus reader. So in a sense, basically the raw activation times were used to create the standard curve, and then we plugged those activation times back into that standard curve that was produced. Now, it's just a fact of linear regression that the line of best fit is, in a sense, going to compromise through those data points to find that line of best fit. And so typically, the extreme ends will be, in a sense, biased in one direction, while the median points will be biased in another direction. And you see that here where in the bottom graph, we're seeing difference from true value for the in-house reader and the SpectraMax ABS Plus. We're seeing, in a sense, an underreporting at the extremes and then an overreporting at the means. That's just the fact that we have a linear regression calculation taking place here. In that line, the data has a slight curve to it, and that line is approximating the regression for that. This is perfectly normal. Once again, we're seeing values well within the 50 to 200% range. That is the overall accuracy, or I should say resolution of the assay. But what I want to focus on is what we're seeing here is difference from true value for these two different readers, but then a comparison of the difference between measurements. And in a sense, we take the values from the SpectraMax ABS Plus reader and make a direct comparison to those endotoxin values from that in-house reader that we would use. You can see virtually no difference between those. Of course, there's variability as we will have in a physical world, but what we're seeing is basically exactly the same values. So what that's telling us is the readers are even picking up on the nuances of the endotoxin assay itself and are providing for us virtually identical results. The important thing, however, is that all test criteria are met for these results, and the difference between readers falls within that measurement error. Of course, the specified error of 50 to 200%, and then also the linear regression error that we were seeing for this specific standard curve. We also did a very similar comparison with the same readers, just with that chromogenic assay, and you can see here the standard curve range now encompasses the entire standard curve that we had, and you can see the activation times. Once again, we're seeing for the SpectraMax ABS Plus reader a slight lengthening of those activation times. You can see the linear regression of the standard curve here that we see. Here you can see the graphs of the time course and the activation that occurred. And you'll notice once again we're seeing that difference in optical density, which is translating for us to that slight difference in activation times. Once again, no necessarily specified requirements for this, just something to observe and take note of. That's what we're seeing here because the readers are going to have different optical features, different ways of measurement. Optical density is a relative value that can vary based on the optics of the physical instruments themselves. But once again, when we plug in those true values and we plug in the activation times against the standard curve to get the measured values, you can see here those differences that are produced, and you can see that linear regression error. The chromogenic reagent performs, in a sense, much better even than the turbidimetric reagent did, but even the difference between measurements lie within those as well. So once again, we see that the conclusion here is that all test criteria are met. We saw that linear regression, the correlation coefficient, absolute value above 0.980, and then all measured values within that resolution of the test. And then even additionally, we see that the differences between the readers are within the error of the linear regression line itself when we plug the values back into each other. So in conclusion here, the Fujifilm Wako endotoxin test reagents and molecular device SpectraMax microplate readers with SoftMax Pro software provide a fully validated solution for your endotoxin testing needs. I want to say thank you for your time and allowing me to bring this presentation. I'm now going to turn the rest of the presentation over to molecular devices, and they're going to explain the details of the software protocol itself, especially focusing on the data compatibility in terms of data compliance with CFR and FDA GMP guidelines for that. So thank you so much for your time.
A senior application scientist at Molecular Devices. In my role with the company, I've worked to validate key assays on our microplate readers to meet the needs of scientists who are doing bacterial endotoxin testing and who need not only a plate reader that can generate the necessary data, but also analysis protocols that allow them to be compliant with regulations. I'm really happy to discuss with you today our collaboration with Fujifilm Wako Chemicals to provide solutions for endotoxin testing. Facts about our microplate readers. Molecular Devices has been building them for nearly 40 years. The picture on the top is the Emax, one of the OG plate readers. In the middle pic, you'll see a SpectraMax reader enjoying some lovely weather in Antarctica. And the bottom photo is the International Space Station, home to two of our SpectraMax M5 readers. So almost anywhere you go, you will find Molecular Devices plate readers. To Earth, where the reader I want to focus on is the SpectraMax ABS Plus. This single-mode absorbance reader has some features that make it ideal for bacterial endotoxin testing. Monochromator-based wavelength selection means that you don't have to install different filters for each assay because you have wavelengths all the way from 190 nanometer to 1000 nanometer, all included on board the reader. Temperature can be regulated from 5 degrees Celsius above ambient to 45 degrees. Of course, this range does take care of the endotoxin assays, which are run at 37 degrees. Kinetic reads are easy to set up, and as I'll show you, we have preset protocols that automate the analysis for Fujifilm's endotoxin assays. The ABS Plus also has 8-channel optics, so 8 wells are read simultaneously. This means that a 96-well microplate can be read in just about 5 seconds. And all of the SpectraMax readers come with SoftMax Pro software. As I mentioned, we provide pre-configured protocols that you can use with the software to analyze assay data automatically. The endotoxin protocols themselves, I would like to talk a little bit about SoftMax Pro software, which is our microplate reader control and data analysis software. Its protocol library has pre-configured protocols for all sorts of microplate reader assays, over 150 protocols in all, including for endotoxin testing, DNA quantitation, ELISA analysis, cell viability, etc. And they're all set up to simplify the data analysis. Compliance tools are available for regulated, which is GLP, GMP laboratories, and we've designed endotoxin protocols that can be used in either regulated or non-regulated spaces. This Molecular Devices software that provides compliance features for GLP and GMP practices, including several features that are really important for labs engaged in bacterial endotoxin testing. With this software, you can achieve full FDA 21 CFR Part 11 and Annex 11 compliance. So for details on how, rather than discussing them here, I'll go ahead and refer you to a Lab Notes article that's on our website. And this lays out very clearly what Part 11 and Annex 11 are, what the responsibilities of regulated customers are, the impact of compliance versus non-compliance, and plenty of other useful information if you're interested. So some of the ways that Softmax Pro GXP software supports compliance are with things like electronic records and electronic signatures that provide audit trails that can be accessed for review. You can also get automatically generated reports to fulfill various compliance requirements. So data are also saved to a secure database, and the database can be backed up and restored. If you'd like more information on how Softmax Pro GXP software can help you keep your lab compliant, please visit our website. There's a wealth of information, articles, white papers, tutorials, videos, etc. Now, part of the endotoxin testing solution that we provided, along with Fujifilm, which is our endotoxin test protocols. These are among our newest protocol offerings, and as such, we do make them available on SpectraNet, which is our customer care portal on Molecular Devices website. It includes our knowledge base, which is a great place to get detailed information, articles, answers to many of your questions. SpectraNet is also where you'll find the Softmax Pro protocol homepage, where you can find the latest versions of protocols that may not have been implemented yet in the Softmax Pro software, but that we want to make available to anyone who would find them useful. This is where you'll find the 2 endotoxin protocols that we've co-designed with Fujifilm to work seamlessly with their kinetic turbidimetric and kinetic chromogenic endotoxin assays. Analysis that can be done with the SpectraMax ABS Plus reader for a kinetic turbidimetric endotoxin assay. It's a 2-hour kinetic read with absorbance reads taken at 405 nanometers every 40 seconds for the duration of the read. This adds up to about 181 data points, giving us plenty of resolution to make accurate calculations from the data. The calculation or the reduction that is done on this kinetic data includes setting the first OD or optical density value to 0, and then calculating the amount of time that's required for the subsequent OD readings to reach a specified threshold or onset OD. So the higher the endotoxin concentration is, the more rapidly this onset OD will be reached. Once we've generated all the data, a standard curve is plotted as mean onset time versus control standard endotoxin or CSE concentration. The curve fit that we've chosen here and that Timothy mentioned before is the log-log fit. This is one of the many curve fits that are available in Softmax Pro software. And for the standard curve for this assay, the correlation coefficient or R value for the standard curve must be at least 0.98. In this example from SpectraMax ABS Plus, you can see that the requirement is met and in fact exceeded. I'm going to walk you through the kinetic turbidimetric assay protocol. This one has been designed specifically for the Pyrostar ESF plate assay from Fujifilm Wako Chemicals. And the first thing you'll see is the introduction, which outlines how the protocol is to be used and also gives information on how the assay is set up. It also describes how to set up the protocol's template so that the samples in the different wells of the microplate will be analyzed appropriately. Instructions on entering information in the reagent info and product summary sections, which I'll show you in a moment, are also given here. Of the protocol are the user entry and the signature section. The user entry section has areas for entry of things like the technician, who's performing the assay, the date the assay was performed, and then various information on the reagents used, including the lot number for each and the expiration date for each. Product information is also entered, and that would include information such as the manufacturer, the product code, product description, product lot number, and the expiration date. There's also an area where assay-specific values, like the endotoxin limit that you need to set for your test, the lowest concentration on the standard curve, and also the product dilution are entered and displayed. Now, the signature section serves as an important record of tester information and test date, as well as approval and the date that it was approved. Securing these details is very important to regulatory compliance. See the product summary section, where statistical linear regression parameters are pulled directly from the standard curve. That's once the assay is run, and calculations are automatically applied so that you can see right away whether your results pass or fail. Over on the right-hand side, you'll see an overall judgment on the assay. Again, this is pass or fail. In this case, the product endotoxin concentration, which is 2.445, has exceeded the sample endotoxin limit of 0.5 EU per mil, so it's clearly indicated as a fail. On the other hand, the positive product control spike recovery was within the acceptable range for the assay, so it's indicated as a pass. Other assay criteria, such as the absolute value of the correlation coefficient, also passed, but all parts of the assay must pass for the overall result to be a pass. It's sections where the data are actually displayed and analyzed, starting with the plate or the test plate section. Here you'll see the instrument settings used to run the assay, and in this section you can also use the template to assign specific wells to groups such as standards, negative control, or positive product control. Once the plate's read, you'll see in the plate view all of the raw kinetic traces, and you can get a general overview of kind of how your assay worked. At the bottom, the reduction settings are summarized, and on the right-hand side is the read information. That's where the parameters like instrument information is displayed, along with the read date, the time, and temperature, things like that. If for some reason you're finding that your results don't look as expected or questions arise later on, having a record like this could be very useful to compliance. My favorite sections, the standard curve. Data for the standards are plotted here as mean onset time versus the control standard endotoxin concentration, with a log-log curve fit automatically applied to the graph. So curve fit results are displayed here, and you'll see some of the parameters that are also summarized in the product summary section that we looked at previously. The R-squared value is also displayed here, and it lets you know how good, objectively, your standard curve linearity really is. Section will list the onset time for each well, along with the mean and the standard deviation and the percent CV. For each set of standard replicates, pass indicates a percent CV that's less than 20. Below the table, you'll see a summary of statistical regression parameters, and there's a calculation to determine whether the absolute value of the correlation coefficient test passes or fails. The sections I'm showing here are for the negative control, the sample, and the positive product control. The negative control should confirm the absence of detectable endotoxin in the assay system, and as you can see here, it does. In the sample section, you get to see whether endotoxin that's detected is under the limit that you've set for your assay. In this example, although the assay replicates are good, you can see the percent CV is less than 20 for each set of replicates. The sample endotoxin concentration has in fact exceeded the test limit, so the overall result is a fail here. The positive product control, or PPC, is a test that's performed to make sure that the product you're testing doesn't have interfering factors in it that are going to skew the results of your test. So in this test, what you do is spike the test sample with a known quantity of endotoxin. As Timothy mentioned, this is typically a concentration of endotoxin that's near the middle of the standard curve's range, and then you confirm that this is accurately quantified. So the test criteria here is that the calculated spike recovery needs to be between 50% and 200% of the actual concentration. In this example, the result is somewhat close, but it does pass. I really hope that Timothy and I have shown you how Fujifilm Wako Chemicals endotoxin test reagents, in concert with Molecular Devices SpectraMax microplate readers and the SoftMax Pro software that powers them, are a fully validated solution for your bacterial endotoxin testing. And I want to thank you very much for your time and your attention.